Today I am going to explain about topic of enzymes. So enzymes are chemical substances. Which chemical substances catalyze the chemical reaction? That means speed up, accelerates the chemical reaction without undergo any permanent change in their structure. These chemical substances is known as enzymes. Enzymes generally proteinaceous substances. That means all enzymes are proteins, but all proteins are not enzymes. So these are biological origin. So the term enzymes originated from Greek word. The term enzyme originated from a Greek word. So meaning enzyme means in yeast. Enzyme means in yeast. So why? Because enzymes were first isolated, discovered from enzymes. That's why enzymes mean is in yeast. The term enzyme coined by Pune scientist. The term enzyme coined by Pune scientist while working on a fermentation process. Then enzymes were first isolated, discovered by Edward Buchner scientist. So Edward Buchner scientist first time isolated enzymes from yeast. That means Zymase enzyme. Edward Buchner scientist was first isolated, discovered enzyme zymase in yeast. So he discovered while working on a fermentation process. So fermentation of sugar. So fermentation of sugar not only by living condition of yeast. Yeast extract could also ferment this sugar. That means yeast extract may contain some chemical substances. These chemical substances could accelerate, speed up or catalyze the chemical reaction, nothing but the fermentation of sugar. Then he called these chemical substances as a zymase enzyme. He was also awarded a Nobel Prize by discovery of this zymase enzyme. Then J.B. Sumner scientist. J.B. Sumner scientist discovered the urease enzyme. Urease enzyme in a Georgina plant. Canovalia. So J.B. Sumner scientist was discovered urease enzyme in Georgina, Canovalia plant. He was also awarded a Nobel Prize. Then Northrop, then Haldane. So Northrop and Haldane discovered the proteinaceous nature of an enzyme. So generally Sumner scientists discovered the crystalline structure of the enzyme, crystalline condition of an enzyme urease. Then Northrop and Haldane scientists discovered the proteinaceous enzymes. So these are pepsin, trypsin, then rhino, trypsin, etc. So Northrop and Haldane discovered the proteinaceous nature of enzyme. Protein enzymes, these are pepsin, trypsin and chymotrypsin. Then Michaelis. So Michaelis, then mention Lineweaver, but then Dixon. So Michaelis, Mention, Lineweaver, Burke and Dixon scientists proposed the enzyme kinetics. Enzyme kinetics. So these enzyme kinetics study the rate of enzymatic reaction and the specific activity of the enzyme. Rate of the enzyme reaction and the specific activity of enzymes. Then enzymes are physically collides. Enzymes are physically collides. Then enzymes are chemically proteins or RNAs. Then enzymes biologically catalyst. So enzymes physically collides chemically proteins RNA and chemically catalyst biocatalyst. So biologically enzymes are synthesized within a cell. That means biological origin. So physically collides then chemically proteins and RNA. That means all enzymes are proteins, but all proteins are not enzymes. Next one, some RNAs also, that means nucleic acids also act as a enzymes. These enzymes is known as a ribozymes. Some nucleic acids also act as a enzymes. So these enzymes is known as a ribozymes. Why? Because some types of RNAs act as a enzymes. This is called ribozymes. So these ribozymes are first discovered by Thomas is a scientist in tetrahymena. Tetrahymena is a ciliate protozoan. Ribosomes was first discovered by Thomas is a scientist in tetrahymena. It is a ciliate protozoan. Example, 23S or RNA. 23S or RNA in prokaryotes. In eukaryotes, 28S or RNA. So generally ribosomes smaller subunit and larger subunit is present. In prokaryote smaller subunit is a 30S. In larger subunit 23 sRNA is present. In eukaryotes in larger subunit 28 sRNA is present. So while that translation process, that means protein synthesis, peptide bondation formation by these enzymes, that means 23 sRNA in prokaryotes, 28 sRNA in eukaryotes by the peptide transferase enzyme. So RNA type is a L19 RNAs. L19 RNA. Some nucleic acids may act as a enzymes. These enzymes is known as a ribozymes. These ribozymes was first discovered by Thomas Setz, scientist in tetrahymena. It is a ciliate protozoan. Best example in prokaryotes 23S or RNA. This 23S RNA present in larger subunit of ribosomes, prokaryotes. Then 28 sRNA in eukaryotes, larger subunit of ribosomes. While translation process, protein synthesis, peptide bond formation between two amino acids, among the amino acids catalyzed by peptide transferase enzyme. Nothing but 23 sRNA and 28 sRNA. Then generally enzymes are synthesizing in a cell, inside the cell. So all components of the cell contain the enzymes, cell wall, cell membrane, etc. But 70% of enzymes are present in mitochondria. That means respiratory enzymes. Major enzymes are respiratory enzymes. So 70% of enzymes are present in mitochondria. So these enzymes are synthesizing within the cell, biological origin, but these may function inside the cell or outside the cell. So based on these enzymes are two types. So these are exoenzymes, then endoenzymes. Exoenzymes and endoenzymes. All enzymes are synthesizing inside the cell, but these may work outside the cell or inside the cell. So based on these characteristic features, enzymes are exoenzymes, that means extracellular enzymes, endo enzymes, intracellular enzymes. Both types of enzymes are synthesizing within the cell. So after synthesis, these may secrete to outside the cell. So why? Because these have to perform enzymatic activity outside the cell because of substrates. Enzymatic substrates are may be present outside the cell. That's why that specific enzymes, which enzymes are responsible to how to digest or catalyze that particular substrates. These move from inside to outside, then perform the catalytic activity. These type of enzymes is known as a exoenzymes, all digestive enzymes. So digestive enzymes. So digestive juice contain these exoenzymes, extracellular enzymes. Example, amylase, then pepsin, lysozyme, etc. So lysozyme in tears, then nozzle, secretions, etc. So lysozyme enzyme ears, nozzle, secretion. Then endoenzymes. These enzymes are synthesizing and performing the catalytic activity inside the cell. These enzymes is known as endoenzymes. So all respiratory enzymes, cellular respiratory enzymes. So enzymes of glycolysis, then Krebs cycle. Glycolysis and Krebs cycle enzymes, these are endoenzymes. The precursor of synthesis of enzymes is known as a zymogen or proenzyme. The precursor to synthesis of enzymes is a zymogen or a proenzyme. So generally this zymogen proenzyme is a precursor to synthesis of enzymes. It is inactive in a condition. Then by cleavage this zymogen and proenzyme to form the active form of enzymes. Then formation of active form of the enzyme depending on specific pH values, then availability of a specific substrate molecule and some special conditions. So for example, the proteinaceous pepsinogen. So proteinaceous pepsinogen enzyme is an inactive form of a pepsin enzyme. So in presence of HCl, this pepsinogen inactive form of a enzyme become active, pepsin become active. So pepsin enzyme. So precursor of enzymes is a zymogen or proenzymes. So like proteins, enzymes also contain secondary structure, tertiary structure and quaternary structure. So if we imagine proteins as a rigid rod like condition, these proteins may have the secondary, tertiary and quaternary structure. So like proteins, enzymes also contain secondary, tertiary and quaternary structure. So why? Because enzymes is nothing but proteins. All enzymes are proteins. So we know very well tertiary structure of the protein is a backbone for the protein. Tertiary structure is a backbone for a protein. So tertiary structure means tertiary structure means the secondary structure of the protein coiled upon itself to form a hollow ball like a structure. This is called tertiary structure. Due to this coiling, foldings, bendings at some places form a crisscross condition. That means crevices, pockets. So these crevices, pockets is known as active sites. active sites. So tertiary structure of protein is a backbone for a protein. This tertiary structure shows some crisscross foldings, crevices, pockets. Each pocket may act as a active site of a enzyme. So active site means at which place on enzymes substrate molecules are bind for catalytic function of a enzyme. This site is known as a active site. Generally, enzymes do not alter the equilibrium state of a chemical reaction. Enzymes do not alter the equilibrium state of chemical reaction. At the same time, while the chemical reaction, enzymes do not undergo any permanent change in their structure. So, generally, enzymes are a biological catalyst, biocatalyst, chemical catalyst. So, these chemical catalyst differentiate with inorganic catalyst. That means some inorganic ions may be act as a catalyst. So to catalyze the chemical reaction. But this chemical catalyst differentiate from inorganic catalyst. So inorganic catalyst. Then chemical catalyst. So generally inorganic catalyst origin is a inorganic condition. So inorganic nature. Then chemical catalyst is a biological origin. Then inorganic catalyst, the shape, size similar to substrate molecule. There is no large variation in size, shape of a substrate and inorganic catalyst. But enzymes have the larger shape compared to larger size and shape compared to substrate molecules. So inorganic catalyst has a lesser variation in shape, size of a substrate and inorganic catalyst, but enzymes are larger in size compared to substrate molecule. So inorganic catalyst generally has a low molecular weight. So molecular weight is a low in condition. Enzymes has a high molecular weight. Enzymes has a high molecular weight. Inorganic catalyst has a low molecular weight. Next one. Inorganic catalysts are less efficient. Inorganic catalysts are less efficient condition. These are the high efficient condition. So inorganic catalysts are less efficient. These are high efficient condition. Then inorganic catalyst can catalyze the reaction at a high temperature and high pressure. High temperature and high pressure. So inorganic catalyst work efficiently at a high temperature and a high pressure. But organic catalyst, chemical catalyst, enzymes do not catalyze the chemical reaction at a high temperature and a high pressure. So in low temperature, so below the 10 degree centigrade, these enzymes are inactive condition. At more than 40 degree centigrade, these enzymes become denatured condition. So inorganic catalyst efficiently work at a high temperature and a high pressure. But this chemical catalyst enzymes at low temperature below 10 degrees centigrade inactive condition, then above 40 degrees centigrade may be denatured condition. But at a high temperature, these enzymes may lose secondary structure, tertiary structure and quaternary structure like a proteins, but primary structure remains same condition, non-altered condition, unaffected condition. But some enzymes isolated from organisms what are surviving, growing at a high temperature, that means hot vents and sulfur springs. These organisms is known as a thermophilic organisms. So what enzymes isolated from these thermophilic organisms? Which organisms are living in high temperature, hot wells and sulfur springs? These enzymes may work efficiently at a high temperature, nearly 90 to 120 degree centigrades. 120 degrees centigrade. So thermophilic stability, high temperature stability is a main property quality of these enzymes. So which enzymes isolated from thermophilic organisms? So best example, Taq polymerase. Taq polymerase enzyme. So these enzyme isolated from Thermus aquaticus bacteria. Thermus aquaticus bacteria. This Taq polymerase enzyme widely used in a PCR technique. So polymerase chain reaction technique for amplification of DNA in recombinant DNA technology. Then PFU polymerase. PFU polymerase. So Pyrococcus furiosus. Pyrococcus furiosus, PFU polymerase. Then Wenge polymerase. Wenge polymerase. Thermococcus litoralis. Thermococcus litoralis. So these enzymes can efficiently work at a high temperature, nearly 90 to 120 degree centigrades. So the main important quality is a thermostability. These enzymes isolated from thermophilic organisms. Enzymes shows the high reaction specificity. Enzymes are highly specific in a reaction. So these are substrate specific condition, reaction specific condition, stereospecificity. So enzyme shows the high reaction specificity. That means specific enzymes work on a specific substrate molecule only. That means catalyze the specific chemical reactions only. So these are substrate specific condition, then reaction specific condition, then stereo specific condition. Enzymatic activity will affect by pH values, then temperature, then substrate concentration, enzyme concentration, then inhibitors, that means chemicals. Enzymatic activity will affect by pH values, temperature, substrate concentration, enzyme concentration and inhibitors, nothing but chemicals. Next one, in a living organism, thousands of chemical reactions takes place. All these chemical reactions, sum of chemical reactions is known as a metabolism. Metabolic pathway. So metabolic pathway is a multi-step reaction. So in this multi-step reaction, each reaction may be catalyzed by same enzyme or different kinds of enzymes. So each reaction catalyzed by same enzyme or different kinds of enzymes. The rate of the enzymatic action, determined by the amount of products are forming. So by the enzymatic action. So the rate of enzymatic action determined by amounts of products. So rate of enzymatic action means what amount of substrates converts into products by enzymatic action is known as a rate of reaction. So rate of enzymatic action. So rate equals to delta P by delta T. So rate of enzymatic action, rate equals to delta P by delta T. Nothing but products by time. So rate of enzymatic action means what amount of substrates are converted into products by enzymatic action is known as a rate of reaction. This rate of reaction determined by amounts of products. Any chemical or physical reaction, any physical or chemical reaction catalyzed by these enzymes. So in living organism, thousands of chemical reactions are takes place. All these chemical reactions are catalyzed by the enzymes. There is no uncatalyzed reactions in an organism. Even the dissolving CO2 in water also catalyzed by living system. So dissolving of CO2 in water catalyzed by living system. So by dissolving here form the carbonic acids. Carbonic acid. So the dissolving of CO2 even in water catalyzed by living system. By dissolving CO2 in water form the H2CO2 carbonic acid. In absence of enzymes as a catalyst, approximately 200 CO2 molecules are dissolved in water from the 200 H2CO3 carbonic acid molecules per hour. But in presence of enzyme carbonic anhydrase. So carbonic anhydrase present in cytoplasm of the cell freely. This carbonic anhydrase enzyme catalyze the reaction, the CO2 dissolved in water. By this dissolving from the H2CO3 carbonic acid. So by catalyze this reaction by carbonic anhydrase enzyme approximately dissolve 6 lakh CO2 molecules in water. So from the 6 lakhs carbonic acid molecules per second. 6 lakhs carbonic acid molecules per second. So that means the enzymes catalyze the chemical reaction millions of times. So approximately 10 million times. So 10 to the power of 7 times. So enzymes increase the rate of reaction. Enzymes increase the rate of reaction approximately 10 million times, 10 to the power of 7. By catalyzing carbonic anhydrase enzyme, 6 lakhs CO2 molecules are dissolved in water to form the 6 lakhs carbonic acid H2CO3 molecules per second. In absence of enzyme as a catalyst, only 200 CO2 molecules are dissolved in water from the 200 H2CO3 molecules per hour. So by catalyzing this reaction form 6 lakhs H2CO3 molecule by the enzyme carbonic anhydrase enzyme. This carbonic anhydrase enzyme freely present in cytoplasm of a cell. So this is known as a turnover number. TON, turnover number. Turnover number means number of substrate molecules converted into products by 1 mole of enzyme, 1 molecule of enzyme per unit of time. So 1 minute, 1 hour or unit of time. This is known as a turnover number. Each enzyme have the specific turnover number. So calculates the turnover number. So number of substrates. Number of substrate molecules are cleaved by enzyme. Then number of enzymes. Number of enzymes are acted into 1 by time. So calculates the turnover number by the formula. Number of substrate molecules are cleaved. The number of enzymes are acted into 1 by time. Substrate molecules is nothing but chemical molecules. So which chemicals bind on active site of the enzyme? By the activity of enzyme converted into products is known as a substrate. So enzymes increase the rate of reaction. Next one, the nature of the product. The nature of the product also depend on reaction, condition and environment, compartment where enzymatic action takes place. The nature of the product, by the enzymatic activity, substrate molecules are converted into products. That means substrates are transformed into products. The nature of the products depending on reaction, condition and environment, compartment where this enzymatic action occurring, that means takes place. So for example, glucose molecule. So glucose molecule generally respiration takes place in a mitochondria. It is an anaerobic respiration, then aerobic respiration. So glucose molecule by the glycolysis converted into pyruvic acid. So CH3COOH, pyruvic acid. So glycolysis takes place in anaerobic condition, so without use of the oxygen. So end product of glycolysis is a pyruvic acid. So these pyruvic acid in absence of oxygen, if oxygen is not available, these may enters into fermentation process. So fermentation is a lactic acid fermentation, then alcoholic fermentation. So lactic acid fermentation in muscles of animals. So in absence of oxygen, this pyruvic acid convert into lactic acid. In lactic acid fermentation, muscles of animals. Then in yeast it is converted into alcohol and CO2. So end product of glycolysis in absence of oxygen may be converted into lactic acid, alcohol and CO2 fermentation process. If oxygen is available, this pyruvic acid enters into mitochondria from cytoplasm. Why? Because glycolysis takes place in cytoplasm of a cell. When move from cytoplasm to mitochondria, then complete oxidation takes place in a mitochondria. Oxidative decarboxylation of a pyruvic acid. So converted into acetyl CoA. Then acetyl CoA enters into Krebs cycle. By the complete oxidation of this acetyl CoA, then form the reduced OA enzymes NADH, then FADH2. Then takes place the electron transport chain, electron transport system. In electron transport system, oxidation of NADH and FADH2. So here finally synthesis of a ATP molecule. So depending on condition, reaction condition, environment, compartment where enzymatic action takes place, maybe different products are possible. Different products may be possible condition. Next one, enzyme shows the regulatory function in a cell. Enzyme shows the regulatory functions in a cell. In regulatory functions in cell, maybe enzymes undergo permanent change in their structure. It is a rare in a condition. This is known as a covalent modification. Covalent modification by the allosteric inhibition. Allosteric inhibition. So covalent modification is nothing but in regulatory function of a cell. These enzymes may undergo permanent change in their structure. This is known as a covalent modification by the allosteric inhibition. So chemical compound generally undergoes to two types of changes. So any chemical compound undergoes to two types of changes. These are physical change, then chemical change. Physical change and chemical change. So in physical change, simply change the shape and state of the compound. Simply change the state and shape of the compounds without breaking existing bonds, without making new bonds, without formation of new bonds. This is known as a physical change. In chemical change, by breaking existing bonds, then make a formation of new bonds, so completely change the shape and state of the structure. So product shape and structure varies with the substrate shape and structure. So physical change. So in physical change, simply change the shape and state of the chemical compound. This is known as a physical change without breaking existing bonds and formation of new bonds. Example, dissolve sugar or salt. Dissolve the sugar or salt in water. By dissolving sugar or salt in water, change the shape only, state of the matter also. Next one, ice. So ice melt to water. Ice melt to water, then water becomes vapor. Water becomes vapor. So ice melt to water, then water becomes vapor. So change the state of the matter. This is known as a physical change. So in chemical change, by breaking the existing bonds, then form the new bonds. So change the shape and state of the matter. Chemical change. So in chemical reactions, inorganic reaction and organic reactions. So BaOH to H2SO4, BaSO4 plus 2H2O molecules. So it is a inorganic chemical reaction. Then organic chemical reaction, hydrolysis of starch. Hydrolysis of starch. So generally starch is a polysaccharide. So it composed by only one type of glucose units, monosaccharides. So beta D glucose units are present in starch. Beta glucose D units are present. So these glucose units are joined by glycosidic bonds. Joins by glycosidic bond. By the hydrolysis of this starch, break the glycosidic bonds, then form the glucose units. Next one, sucrose. Sucrose is a disaccharide. That means two types of monosaccharides are present. So these are glucose, then fructose. Glucose and fructose. This glucose and fructose joins by glycosidic bond. So hydrolysis of these sucrose by sucrase enzyme break the glycosidic bonds, then form the monosaccharides, glucose and fructose. Glucose and fructose. So two types of changes in chemical compound, physical change and chemical change. Next one, nature of enzyme action. Nature of enzyme action. Nature of enzyme action. So each enzyme has a specific site for substrate molecule. This site is known as a active site of enzyme. Then substrate molecules bind on a active site of enzyme. So form the enzyme substrate complex. So each enzyme has a specific site for specific substrate molecule. Then substrate molecule diffuse towards the enzyme, then bind on active site of enzyme, form the enzyme substrate complex. So this enzyme substrate complex is a short-lived condition, transient state of the structure, temporary state of the structure. Then here form the enzyme product complex enzyme. That means in enzyme substrate complex break the existing bonds, then make a formation of new bonds. So why this time form the enzyme product complex? Then enzyme release the product. So enzyme is a free in condition without undergo any permanent change in structure of the enzymes. Free enzyme. So enzyme product complex between the product and the enzyme substrate complex. Then free enzyme ready to bind with another substrate molecule, then ready to do another chemical cycle. So each enzyme has a specific site for substrate. This is known as a active site. Then substrate molecule bind on active site of enzyme from the enzyme substrate complex. Then from the enzyme product complex, then release the product. Free enzyme ready to bind with another substrate molecule. So in this first action is substrate molecule diffuse towards the enzyme, then bind on a active site of a enzyme, then substrate molecule fit tightly on a active site of the enzyme, then substrate molecule induces on active site of the enzyme to alters its shape for more tightly bind or fit with the enzyme. That means on active site of a enzyme. Then active site of the enzyme undergoes to some conformational changes for more tightly fit or bind with the substrate molecule. So from the enzyme substrate complex. Why this time expecting bonds will break, then make the new bonds from the enzyme product complex, then release the product. So free enzyme ready to bind with another substrate molecule, then do the another chemical cycle. If we plot a graph for this enzymatic action, if we plot a graph for this enzymatic action, so y-axis, then x-axis. So x-axis represents progress of a reaction, enzymatic reaction. Then y-axis shows the potential energy. What amount of energy available in substrate molecule. So y-axis shows the potential energy, x-axis shows the progress of a reaction. Then while this time substrate molecule. So substrate molecule diffuses towards the enzyme, then bind on a active site of a enzyme from the enzyme substrate complex. It is nothing but enzyme substrate complex. So this enzyme substrate complex is a short-lived condition, transient state of the structure. So temporary state of the structure. Then from the enzyme product complex, then release the products. So free enzyme without any alters in shape, ready to bind with another substrate molecules. So enzyme substrate complex is a transient state of the structure, then form the product. If a product is below the substrate molecule, the energy amount of the products below the substrate molecule. This type of reaction is known as a exothermic reaction. Exothermic exorganic reaction. If a product below the substrate molecule, this type of enzymatic action is known as a exothermic reaction, exorganic process. That means energy releasing process. Let's take example, respiration. We need not to provide the energy for this type of reactions. So why? Because it is an energy releasing reactions. Energy levels are low in condition. It is a spontaneous reaction. It's depending on availability of raw materials for respiration. If a product is greater than the substrate molecule, that means above the substrate molecule, this is known as endothermic reactions. Endothermic reactions. Endothermic means uptake the energy from its surrounding, energy accepting reaction, endothermic reaction. So any type of reaction, exothermic reaction or endothermic reaction, must pass through the enzyme substrate complex. That means a transient state of the structure. Any type of the reaction, exothermic spontaneous reaction or endothermic reaction, energy uptaking, energy releasing reactions must pass through the transient state of the structure. So while these structures, enzyme substrate complex, then product form the number of altered states of the structures. So between these states of the structure, substrate enzyme complex, substrate product complex, then products form the number of altered states of the structure. But all these states of the structures are unstable condition. All these states of structures are unstable condition. So stability refers something related to energy status. That means in unstable altered structures, energy levels are low in condition. So because of this, these are unstable condition. So stability refers to something energy levels. Then here enzyme with activation energy with the enzymes. So it is activation energy without the enzymes. So generally enzymes accelerate, speed up the chemical reaction, do not alter the equilibrium state of the structures. So enzymes increase the rate of reaction, decrease the activation energy. So activation energy means the difference in average energy of substrate and enzyme substrate complex. This is known as activation energy. Nothing but what amount of energy required to form a enzyme substrate complex for the enzymatic action is known as activation energy. So what amount of energy requires to become initiates the enzymatic action is known as activation energy. So activation energy means the difference in average energy of substrate and enzyme substrate complex, transient state of the structure is called activation energy. So enzymes always increase the rate of reaction, speed up, accelerates the action, decrease the activation energy. So by consuming less amount of energy, it speed up, accelerates the chemical reaction. So nature of enzyme action. So any reaction, exothermic and endothermic, must pass through the transient state of the structure. That means enzyme substrate complex. So enzyme substrate complex is a transient state of the structure. So next one, coming to mechanism of enzyme action. Next one, mechanism of enzyme action. So mechanism of enzyme action explained by two hypotheses. So first one, lock and key hypothesis. Second one, induced fit hypothesis. Induced fit hypothesis. So mechanism of enzyme action explains by two hypotheses, lock and key hypothesis, then induced fit hypothesis. So first one coming to lock and key hypothesis. So lock and key hypothesis proposed by Emil Fischer. Emil Fischer scientist, but these are developed, widely developed by Fielders, then Wood scientist. So Fielders and Wood scientist developed this lock and key hypothesis. According to this hypothesis, active site of the enzyme is a residue. Active site of the enzyme is residue, not flexible condition. Not flexible. According to lock and key hypothesis, active site of the enzyme is a residue condition, so not flexible in condition. Then active site of the enzyme may contains a number of geometrical shapes with some chemical groups. So these groups are NH2, COOH and ASH groups, geometrical shapes along with these chemical groups. These geometrical shapes similar with geometrical shapes of substrates. That means enzymes are substrate specific condition. So what geometrical shapes are present on active site of the enzyme? These are similar to geometrical shapes of a substrate molecule. Then by this one, substrate bind a specific enzyme, then perform the activity, then form the enzyme substrate complex, then enzyme product complex, then release the product, free enzyme ready to bind with another substrate molecules. So we can compare this one with lock and key. So key is nothing but substrate then lock enzyme. So we can compare this one with lock and key. So in this case, lock is nothing but enzyme. So inside the lock may contain some geometrical shapes. So these geometrical shapes are similar with lock. So nothing but substrate molecule. Then substrate molecule. So bind with this lock. So substrate molecules binds with a lock, then form the enzyme substrate complex, then enzyme product complex, then release the product. That means we can open a specific lock by the specific key only. The opened condition is nothing but a product. So this key has some geometrical shapes. These are similar with the geometrical shapes of active site of the enzyme or a lock. This is known as a lock and key hypothesis. According to lock and key hypothesis, active site of the enzyme may contain some geometrical shapes. These shapes are similar with the shapes of substrate molecules. So we can compare this one with lock and key. So lock is nothing but enzymes, key is nothing but substrate. So inside the lock a specific geometrical shapes are present. These geometrical shapes are similar with the key. So we can open specific lock by the specific key only. So enzymatic action also similar to relationship of lock and key. So that's why this is known as a lock and key hypothesis. According to this hypothesis, the active site is a very rigid condition, not flexible in a condition. Next one, induced fit hypothesis. So induced fit hypothesis proposed by Daniel E. Covalent scientist. Daniel E. Covalent. Induced fit hypothesis was proposed by Daniel E. Covalent scientist. This hypothesis is a modification of lock and key hypothesis. This hypothesis is a modification of lock and key hypothesis. According to this hypothesis, active site of the enzyme is a not rigid condition. Not a rigid condition. So it is a flexible. So flexible condition. Active site of the enzyme is a not a rigid condition. So it is a flexible in condition. But in lock and key hypothesis, active site of the enzyme is a rigid, non-flexible condition. Here non-rigid condition. So it is a flexible in a condition. Enzyme buttressing group, then catalytic group. Active site and substrate, induced fit hypothesis. So active site of a enzyme. So active site of the enzyme is a non-rigid condition, may be flexible condition. So active site of the enzyme may contain two groups. So these are buttressing groups. So these buttressing groups provide support to substrate molecules. So buttressing group provide support to substrate molecule. Next one, catalytic group. So this catalytic group so perform the catalytic action. This active site of the enzyme also contain amino acid residues as a functional groups. Amino acid residues as a functional group. According to induced fit hypothesis, active site of the enzyme is a not rigid, flexible in a condition. This active site of the enzymes may contain two groups. So buttressing group. This group contain amino acid residues as a functional groups. This buttressing group provide support to substrate molecule. Then catalytic group. So after form the enzyme substrate complex. break the expecting bonds, then make the new bonds. This is called catalytic group. Next one, substrate. The shape of the substrate molecule, the active site of the enzyme is a variable condition. That's why active site of the enzyme is a flexible in a condition. So initially substrate molecule diffuse towards the active site of a enzyme, then bind on a active site of a enzyme loosely, not a rigid condition. Why? Because the shape of the active site and substrate molecule shape is a variable condition. That's why initially from the enzyme substrate complex loosely form, then the substrate molecule induced to active site of the enzyme alters to its shape. So while this time this buttressing group provide support to the substrate molecule, then active site of the enzyme undergoes to some conformational changes. By these conformational changes maybe change it shape of the active sites according to regarding to shape of the substrate molecule, then bind tightly with the substrate molecule. Then catalytic group, so break the expecting bonds in substrate molecule, then make or form the new bonds, then form the enzyme product complex, then release the products. Enzyme do not undergoes to any permanent change in structure. So free enzyme ready to bind with another substrate molecule, do again once again chemical reaction. So enzyme mechanism of enzyme action.